All right, let's get right into it. Today, we are cracking open one of the most critical, high-scoring topics for your CUET exam, the secrets of DNA replication. Seriously, if you want to lock in some guaranteed marks, you need to stick with me right to the very end. Okay, let's do this.
Just stop and think about that for a second. 3 billion letters of code, copied almost perfectly thousands of times a second in your body. How on earth is that even possible? It's this incredible piece of biological machinery, and, you guessed it, CUET loves to test you on how it works.
And listen, when I talk about marks, I'm not exaggerating. Mastering what we cover in the next 10 minutes or so can absolutely secure you 1 to 2 guaranteed questions. That's a potential 5 to 10 marks added directly to your score. This isn't just learning a topic, it's a strategic play for a better rank.
So, why is CUET so obsessed with DNA replication? Because it's a gold mine for questions to test your core concepts. It is easily the most frequently tested topic in this unit. They're going to hit you with questions on the sequence, they're going to try to trap you at the whole leading versus lagging strand confusion, and they love making you match the right enzyme to its job. We're going to cover all of it, the Meselson Stahl experiment, the tricky assertion reason questions, everything.
Okay, first things first, let's start with the master blueprint. This is the single biggest idea you need to understand. Everything else we talk about today is built right on top of this one core concept.
The whole process is called semi-conservative replication. And you know what? The name is a dead giveaway. Semi means half, right? And conservative means saved. So put it together, in every new DNA molecule that's made, one strand is the original old one from the parent, and the other strand is brand spanking new. Half of the original is literally conserved. Simple as that.
But how do we actually know this is true? Well, thanks to a seriously clever experiment by Meselson and Stahl, here's how they did it. First, they grew some E. coli in a special soup containing heavy nitrogen, N¹⁵. This made all the bacteria's DNA heavy. Then they took those heavy bacteria and moved them into a new soup with normal, light nitrogen, N¹⁴, and let them divide just once. When they spun this new DNA in a centrifuge, they didn't see a heavy band or a light band. They saw one single hybrid band right in the middle. And the final proof? They let it divide one more time, and boom, they got 2 bands, one hybrid and one light. This was the slam dunk evidence that proved the semi-conservative model.
Okay, quick pop quiz. Let's see if you were listening. After that very first round of division in the light, N¹⁴ medium, how many bands did they see in that test tube? Go ahead, pause for a second and lock in your answer.
Did you get it? The answer is just one single hybrid band, sitting perfectly between where a heavy and a light band would be. If you got that right, fantastic. You've just understood the core proof of this entire process.
So we've got the blueprint. Now let's meet the crew, the workers who actually get the job done. Think of it like an elite construction team of enzymes, where each one has a very specific, very important job. Here's your essential toolkit for the exam. First up is Helicase. I call it the unzipper. It just unwinds that tight double helix. Next, Primase comes along and lays down a tiny little RNA primer. It's like the starting block for a race. Then comes the star of the show, DNA polymerase. This is the builder, adding all the new nucleotides. But, and you need to circle this, highlight it, tattoo it on your brain, it can only build in one direction, from 5' to 3'. That is a huge point for CUET. Finally, we have Ligase, which is the glue that sticks DNA fragments together, and Topoisomerase, which keeps the whole thing from getting tangled up.
Now that one little rule we just mentioned, that DNA polymerase can only build from 5' to 3', creates a really interesting problem. The 2 strands of DNA run in opposite directions. They're anti-parallel. So the cell has to be clever and copy them in 2 totally different ways. This creates what I call 2-speed replication.
Okay, look at your screen. Let's break this down side by side. The leading strand on the left, that's the easy one. It's made smoothly, continuously, just following the helicase as it unzips. It only needs one primer to get started, smooth sailing. But now look at the lagging strand on the right. This one's the tricky one. To obey that 5 to 3 rule, it has to be built backwards, away from the unzipping fork, in little chunks. These chunks are called Okazaki fragments. Each little piece needs its own primer, and later on they all get stitched together. This difference is a classic CUET trap, so make sure you've got it down cold.
All right, the theory is done. Now it's time for the most important part, turning that knowledge into actual marks on your CUET paper.
This is where we shift gears and focus on beating the exam itself.
To help this all stick in your head during a high-pressure exam, I've got a few memory tricks for you. For the main enzyme team, just remember HE-PPL. That's Helicase, Primase, Polymerase, Ligase. For the strands, think leading is logical because it's continuous, and lagging is late because it's made in fragments. And for that all-important direction, just tell yourself 5' is fine. Synthesis is always fine going from 5' to 3'.
Okay, time to practice. Question 1 is a straightforward MCQ. DNA replication is A, B, C, or D. Pause if you need to. And the correct answer is, of course, C, semi-conservative. We know this because each new molecule saves, or conserves, one of the original strands.
Let's try a match the following question. You will see these on the CUET. Let's do it together. A is helicase. What does it do? It unwinds the DNA, so A matches with 2. Easy. B is ligase. Remember our trick? It's the glue. It joins fragments, so B matches with 1. That leaves C, primase, which makes the RNA primer, so C matches with 3. The correct set is A2, B1, C3. How'd you do?
All right, the assertion reason question. These can be tricky, but here's the magic trick. First, read the assertion. Replication is semi-conservative. Is that true? Yes, it is. Now read the reason. Each daughter DNA has one old and one new strand. Is that true? Yes, it is. Now for the final step. Connect them with the word because. Is replication semi-conservative because each daughter has one old and one new strand? Absolutely. That's the very definition. The reason perfectly explains the assertion, so the answer is A.
Okay, everybody, listen up. I'm going to save you from losing easy marks right now. These are the 5 biggest traps students fall into. Number 1, confusing the direction. Say it again, 5' to 3', always. Number 2, mixing up primase and polymerase. Primase starts with RNA, polymerase continues with DNA. 3, forgetting the Meselson Stahl result. Generation 1 has 1 hybrid band. 4, thinking Okazaki fragments are everywhere. Nope, they are only on the lagging strand. And 5, confusing the builder with the gluer. Polymerase builds, ligase joins. Avoid these mistakes, and you are golden.
All right, final 2-minute drill. Let's lock this in for good. The model, semi-conservative. The proof, Meselson Stahl. The main builder, DNA polymerase, which goes 5 to 3. The unzipper, helicase. The 2 strands, leading is continuous, lagging is discontinuous with Okazaki fragments. And the glue that sticks them together, ligase. Got it? Good.
And now for the ultimate challenge question. This is to test if you truly understand the concept. Imagine if replication was fully conservative, meaning the original DNA double helix stayed together and a totally new one was made from scratch. What would Meselson and Stahl have seen after that first generation? Think about it. They would have seen 2 completely separate bands, one all-heavy band, which was the original DNA, and one all-light band, which was the brand new one. If you figured that out, you're in fantastic shape for the exam.
So, how did you do on those 3 practice questions? Be honest. Drop your score out of 3 in the comments below. If you got a perfect score, that's awesome. If you missed one, that's okay too. Now you know exactly what to go back and revise. Mastering topics like this is how you become a top scorer. Keep up the amazing work, and I will see you in the next one.